Hello, welcome back. In the second session, I will tell you how to design and develop your own sensor. That is, design and development of nanomaterials-based biosensors for biochemical applications. How are nanomaterials used in biosensor fabrication? For example, you can, at the end of the lecture, you will be able to develop in your lab your own biosensor for any disease such as even coronavirus. For example, in cancer disease, there are several proteins are overexpressed. For example, PSA, EGFR, these are proteins are overexpressed in cancer. Therefore, by measuring the level of these proteins, we can monitor the progress of the disease by using biosensors. Biosensors are also used for following the therapeutic treatment of the drug. For example, cancer in the case of cancer patient, these biomarkers are found in the blood. Okay, so they are detected and they are measured. The levels are high. After treatment, after they undergo treatment, the levels are decreasing. We can measure the levels of these proteins by using biosensor. Thereby, we can follow the therapeutic process, and also we can whether the drug is effective or not. It is involved in drug development, drug discovery. In this second session, I will share with you the biosensors we have developed in our lab. For example, we have developed small molecule biosensor. Small molecules such as superoxide anion radical, nitric oxide metabolites such as NO, NO2 minus, NO3 minus, cotinine and glutamate. And also we have developed biosensors for proteins, enzymatic biosensors, and we have also developed immunosensors based on antigen-antibody interactions. We have also developed molecular imprinting based biosensors for neuronal disease biomarkers. In one of our recent project sanctioned by SERB DST 23 lakhs, we are developing biosensors, MIP based biosensors for neuronal disease biomarkers. We have also got patent for Cysteine Biosensors. So I will share with you one by one the biosensors we have developed. At the end of the lecture, as I said, you will be able to develop your own biosensor. So kindly follow the lectures very carefully. For example, in this slide, I have shown how we have developed the superoxide anion radical biosensor. What are superoxide radicals? Superoxide radicals are nothing but oxygen O2 when it is impartially reduced. When it is partially reduced, it will give superoxide radical. For example, in the case of hypoxia, as I said earlier, at high altitude the level of oxygen is low, and also during heart attack, ischemia, heart attack, superoxide radicals are produced in large amounts, causing heart damage. So these are all small bullets. How they are detected? These are biomarkers of oxidative stress as well as hypoxia and also heart disease, etc. So how they are detected? For example, in this case, we have used the enzyme superoxide dismutase enzyme. It is very specific towards superoxide radical. It dismutates superoxide to hydrogen peroxide and oxygen. It eliminates superoxide radicals. SOD1 superoxide dismutase enzyme is present in our cells, in our body. It is an antioxidant enzyme. It scavenges the superoxide radical. It specifically scavenges superoxide radical. Now I will tell you how it is fabricated. We have taken platinum electrode. In our early research, we have used platinum electrode. In later part of our research, we have used screen printed electrode for portable applications. For example, in this case, we have taken platinum electrode and we have electropolymerized pyrrole to give polypyrrole, and later on the polypyrrole electrode we have electrode deposited gold nanoparticles, and then we have formed cysteine. In the presence of cysteine, it forms self-assembled monolayer. You see, it forms a SH bond through SH bond, and because of the high stability of the gold and sulfur bond, it will form a self-assembled monolayer, and then using glutaraldehyde we have bioconjugated superoxide dismutase enzyme to the electrode. See, if you simply place SOD1 on the platinum electrode, the SOD1 will not bind to the electrode tightly. Therefore, we have modified the electrode using conducting polymer. Polypyrrole is a conducting polymer, and we have used in the polypyrrole nanomatrix, we have used gold nanoparticles which are impregnated in the cavities of polypyrrole matrix, and again self-assembled monolayer is formed with the thiol. All thiols form self-assembled monolayer. These have groups, functional groups, to be conjugated to the enzymes using glutaraldehyde, as I shown biofunctionalization. Now the biosensor, this is the superoxide radical biosensor we have fabricated. And what are the advantages of gold nanoparticles? We have used gold nanoparticles to immobilize, to bioconjugate the superoxide radical enzyme. Large surface to volume ratio, increased surface activity, and electrical properties. They have high conducting power, therefore they have high current signal. In this, I have shown the working mechanism of superoxide radical biosensor using SOD1 enzyme. This is an enzymatic biosensor, because we have used enzyme as the biorecognition element. We have used the enzyme as the biorecognition element. Actually, at the electrode surface, at the electrode, as you see, copper 2 plus is reduced to copper plus by superoxide radical to give O2, and again with the copper plus, another superoxide molecule Cu plus O2 minus react to give hydrogen peroxide, and it gives back Cu2 plus S superoxide dismutase. At the active site, SOD1 enzyme, superoxide dismutase enzyme, it has got copper metal at the active site. It redox cycles between copper 2 plus and copper plus during the superoxide scavenging. As you see, because of the redox cycling, because of the transfer of electron, when it undergoes oxidation or reduction, there will be current flowing in the electrode. Because of the electrode redox process, there will be current flowing. The current increases. The current, for example, here we have used cyclic voltammetry. We have used cyclic voltammetry like optical method. You will measure the current signal, whereas in optical spectroscopy you will measure the absorbance of analyte by the analyte light absorption, whereas here the electric current is measured in the case of current versus voltage. The voltage is swept. When the voltage is swept from 0.5 to minus 0.5 naught, the current response is followed. For example, we have fabricated the biosensor, and we have confirmed the SOD1, the presence of SOD1 on the electrode surface. This is due to the copper and copper 2 plus irreversible peak is obtained. This peak is due to SOD1. Therefore, we have confirmed that the biosensor is formed. We have characterized. This is a CV is used to characterize the biosensor electrode. Here I have shown the electrochemical response to superoxide radical. We have taken the biosensor, superoxide radical biosensor electrode, and we have added superoxide radical. As you see, we have got huge increase in the current, because of the superoxide radical added to the electrode, added on the electrode. Here we have shown the effect of GNP. In the absence of GNP, the current signal is low. As you see, the current signal is low. Whereas in the presence of GNP, in the absence of without GNP, we have used polypyrrole electrode and immobilized SOD1, and the current response is measured for superoxide radical. Whereas in the presence of gold nanoparticle, the current response is increased 2 to 3 fold. Here we have generated superoxide radical. We have generated the superoxide radical by using xanthine oxidase enzyme. In the presence of xanthine oxidase enzyme gives superoxide radical. With the increase in xanthine concentration, the superoxide radicals formed are greater. Therefore, with the increase in xanthine concentration, the superoxide radicals are increased. For example, 5 micromolar of xanthine, 500 micromolar of xanthine. Therefore, the current response increases because of the formation of high concentrations of superoxide radicals. We have drawn a calibration plot with the increase in the superoxide radical concentration and current. There is a linear relationship between the concentration and current response. In this slide, I have shown you the active site of copper zinc superoxide dismutase enzyme. As you see, there are two metals at the active center of the copper zinc SOD superoxide enzyme Cu2 plus and zinc 2 plus. It is surrounded by histidine. The active site, if you look at it is a huge protein, around 36 kilodalton. Okay, now the active site, if you look at the active site of the enzyme is very narrow. It allows only small biomolecules to reach the active site. Other large molecules cannot penetrate through the active site channel. Therefore, only superoxide radical, nitrite, NO, very small molecules can only pass and react at the copper active site. Using this principle, we have developed a biosensor for DRDO. The simultaneous determination of superoxide radical and NO2 minus, because superoxide radicals are hypoxia biomarkers. Similarly, NO2 minus are also NO2, NO, they are also hypoxia biomarkers. Using the same enzyme, we have simultaneously determined both the analyte. Using the SOD1 modified biosensor, we have simultaneously measured superoxide radical and nitrite ion. As I said earlier, these are small molecules can pass through the narrow active site channel and they undergo redox reaction. They undergo redox reaction at the copper active site of SOD1. For example, superoxide radical undergoes redox reaction at 0, E equal to 0 volt, and whereas nitrite ion occurs at 0.6 volt. Therefore, these two redox signals are well separated. Therefore, we are able to simultaneously measure both the superoxide radical and nitrite ion in the sample, in the blood sample, in the tissue sample, or in the cell sample. In this, we have applied the SOD1 biosensor for the measurement of superoxide radical and nitrate ion released from the cancer cells, MCF7 cancer cells. We have measured this concentration of superoxide radical and nitrate ion in cancer cells and published the results in biosensor and bioelectronics. For DRDO, we have developed a biosensor for nitric oxide metabolites for the determination of nitric oxide metabolites, as these levels are very low in hypoxia patient or hypoxia disorder military jawans. In this, we have developed Enzymatic Biosensor. What do you mean by Enzymatic Biosensor? We have bioconjugated both the enzymes to the same electrode. Earlier, we have biofunctionalized the modified electrode with SOD1. In this case, we have used two enzymes, SOD1 and nitrate reductase enzyme. They are biofunctionalized, bioimmobilized on the modified electrode surface. Therefore, we are able to determine both the nitrate, it occurs in this range, and nitrite occurs in this lower potential range. Therefore, these two peaks are well separated. Therefore, they can be determined by using the nitric oxide metabolites was determined by using Enzymatic Biosensor. In this, we have used biomimetic biosensor. For example, in most of the enzymes or in proteins, metalloporphyrins are at the active site. For example, Cytochrome reductase you will be seeing in next two slides. These metalloporphyrins are present in the active site of many enzymes, Cytochrome reductase, Cytochrome peroxidase. There are several enzymes. Therefore, we have used the manganese metalloporphyrin as enzyme-mimetic biosensor for the determination of nitrite and nitrate. Similar to SOD1 and SOD1, here we have used the metalloporphyrins and determined the and measured the levels of nitrite and superoxide radicals. As I said, nitric oxide is an important biomarker for hypoxia. Okay, so I was asked by the DRDO to develop biosensor for nitric oxide. Therefore, we have in another DRDO sanctioned project, we have developed nitric oxide biosensor, because at high altitude exhaled breath NO level is reduced than normal for hypoxia patients, and it leads to various respiratory diseases. Even in the case of asthma patients, exhaled NO is higher. They have higher NO. They have breathing trouble because of higher NO concentration in their breath. Whereas in the case of hypoxia patient, they have reduced level of NO. Okay. So there is only one method available presently for detection of NO that is chemiluminescence is commonly used in DRDO, and grease method. Grease method is an optical method using optical UV visible spectrophotometer. These are all not portable. So we need to measure the nitric oxide and nitrate levels at high altitude for our military jawans. How the levels vary when you go to hill, when you are in the ground normal sea level. Here we have developed a biosensor for nitric oxide for DRDO. In this we have used the same SOD1 modified electrode as superoxide radical biosensor. Okay, here NO is detected, NO is oxidized and to NO2 minus, NO3 plus. Due to the redox reaction, there will be flow of electrons. These electrons will be converted into electrical signal, and the concentrations of nitric oxide is thus measured. The biosensor electrode fabricated were studied by scanning electron microscope. This is the image of bare platinum electrode. This is the image of platinum electrode after electropolymerization of pyrrole conducting polymer. We have nanoporous matrix. Here, as I said, the surface is smooth. The enzymes cannot bind to the electrode. The enzymes cannot bind to the electrode, whereas the enzymes can bind to this surface because the surface is now very porous. And the porosity and nano matrix is formed by a CNT modified polypyrrole platinum electrode. We have at each step we have characterized the electrode. For example, A is the PPY platinum electrode, PPY platinum electrode, and B is CNT modified electrode, and SOD1. This is SOD1 modified electrode. You could see a peak because of the immobilization bioconjugation of SOD1. So we have characterized the fabrication of biosensor electrode at each step, whether the enzyme is immobilized or not. Here we have increased the concentration of NO and measured the cyclic voltammogram. The current signal increases, as I said, due to increased redox reaction, increased flow of current. There is a linear calibration curve. From the linear calibration curve, for example, if you know for unknown sample, for a biological sample, you know the current, you know the peak current, then from the peak current, and you can calculate the nitrite NO level present in the blood sample or cell sample or even breath sample, etc. In the case of biological samples, there are several interferences. For example, when you measure nitric oxide, they undergo redox reaction. There are other biological substrates, for example, ascorbic acid, uric acid, cysteine, and other amino acids and other biological substrates. They also undergo redox reaction at the electrode. Therefore, there are a lot of interferences. Even in our life, day to life, there are a lot of interferences. Similarly, there are interferences very much in biological samples. Therefore, we have used Nafion membrane for example, even in a glucometer, the glucose oxidase modified electrode is covered with the Nafion membrane or any membrane, so that only glucose can pass through the membrane. Okay, not the blood. When you place a drop of blood on the glucometer, the blood will stick to the glucose oxidase. Once it will stick to the enzyme, the enzyme activity is not in the enzyme will not be active. Therefore, similarly, in our case, we have coated the biosensor electrode with the Nafion membrane. These Nafion membranes are negatively charged. Therefore, ascorbate, urate, they are also negatively charged. So they are not able to pass through the membrane. Therefore, the interferences are eliminated by using after Nafion membrane coating. DRDO scientists have investigated that intake of beetroot juice reduced the hypoxia disorder of military personnel at high altitude, because beetroot is rich in nitrate. They contain 100 micromolar of nitrate. Therefore, during hypoxia, nitrate level is reduced in serum. We have used our biosensor and measured the nitrate levels are reduced in serum of hypoxia disorder military personnel. Okay, so after administration of nitrate rich fruits, vegetables, for example spinach, the nitrate level increases. Therefore, it reduces the hypoxia disorder. How nitrate is metabolized? It is very interesting that we don't have any enzyme to metabolize nitrate. Therefore, it is metabolized by bacteria in the oral cavity that reduce nitrate to nitrite and nitrite to nitric oxide. In gastric acidic non-enzymatic reduction of nitrate to NO occurs. Therefore, nitrate, when you take beetroot juice, nitrate is converted into nitric oxide. Therefore, it increases the level of nitric oxide, and hence there will be no disorder, hypoxia disorder. It gives relief from hypoxia disorder. It is used for therapy of hypoxia disorder. Here, nitrate was determined using nitrate reductase based biosensor. As I have shown here the construction of nitrate biosensor. It is similar to superoxide radical biosensor, SOD1 biosensor. We have coupled the electrode, bioconjugated the electrode with the nitrate reductase, and we have measured the nitrate level by using nitrate reductase. During the enzyme actually reduces nitrate to nitrite. Due to the redox change, the current response varies. Here I have shown the bare electrode, PPY modified electrode. These are all gold nanoparticles. We have electrode deposited the electrode surface with the PPY modified surface with the gold nanoparticle to conjugate nitrate reductase. We have determined the nitrite levels in beetroot supplement provided by DRDO, and we have also determined the nitrate concentration determined by biosensor method, and we have also determined by grease method using optical method. They agree with each other. So far we have developed biosensor for small molecules. In this we have developed biosensor for cytochrome C protein. It is a heme containing metalloprotein. We have received 32 lakhs from DPT, New Delhi, for the development, for the design and development of Cytochrome C biosensor. Cytochrome C is a very important protein. It is a heme containing metalloprotein. It is located in the intermembrane space of mitochondria. I have shown here the 3D structure of cytochrome C. The prosthetic group is heme. As I said, heme is present at the active site. It is involved in electron transfer process in the mitochondria. Cytochrome C is an important protein involved in cell death in cell apoptosis. Apoptosis is called cell death, cell suicide. Cytochrome C is the major essential component in the electron transfer chain. It settles electrons between cytochrome C reductase complex 3 and cytochrome C oxidase complex 4. In the electron transfer chain, oxygen, for example, oxygen is completely reduced to water to give water by means of 4 electron reduction. This reduction is 4 electron reduction to give water. If it is partially reduced, for example, in most of the neuronal diseases or in cardiac diseases, etc., these are because of even in hypoxia, the oxygen is not completely reduced. The oxygen is not completely reduced. The oxygen is partially reduced to give superoxide radical. This superoxide radical is dismutated to hydrogen peroxide, causing extensive damage to cell, heart, leading to heart attack, etc. Here, how cytochrome C is involved in apoptosis, cell apoptosis. For example, during oxidative stress, the cell membrane is damaged, and cell mitochondria, cell components, cell organelles were damaged. Cytochrome C is released into cytosol. Therefore, the cytochrome, once apoptosis is initiated, the cytochrome C level increases in the cytosol. Okay, by measuring the cytochrome C in cytosol, we can measure the cell death. There are two types of cell death, apoptosis and necrosis. There are two types of cell death: apoptosis, normal cell death, cell suicide, cell after long life, the cell commits suicide and it kills itself. Whereas necrosis is due to, for example, accident or heat damage or other external damage. It causes cell death because of cell damage. So cytochrome C is the biomarker of apoptosis, as we have seen earlier. We have cytochrome C released from mitochondria to cytosol under pathological condition. In cytosol, it further activates cascades leading to cell death. So, the measurement of cytochrome C in cytosol used as the marker of apoptosis. So, we have applied for a project to Department of Biotechnology for the development of cytochrome C biosensor using cytochrome C reductase. There are two possibilities because cytochrome C in electron transfer chain in mitochondria, it settles electrons between cytochrome C oxidase and cytochrome C reductase. There are reports who have used cytochrome C oxidase to measure cytochrome C level, and in our study we have used cytochrome C reductase to measure the oxidized form of cytochrome C that is actually involved in apoptosis, because in oxidized form only it is released into the cytosol. Here I have shown the fabrication of cytochrome C biosensor using cytochrome C reductase. Using cytochrome C reductase, the the, the fabrication steps are very similar as as I said in first sensor. So we have used platinum electrode. We have bioconjugated. We have conjugated the electrode, modified electrode surface with the cytochrome C reductase in this case. This is the working mechanism of cytochrome C biosensor. What are the redox process taking place? It involves cytochrome C involves in redox cycling between Fe3 plus and Fe2 plus. It is mediated by hydroquinone or any quinone. We have published this result in biochemistry journal, bioelectrochemistry journal. Here I have shown the role of gold nanoparticle and CNT, and this is CNT modified electrode cytochrome C current response, and this is current response for gold nanoparticle, and this is without any nanoparticle. As you see, in the presence of nanoparticles, the current response is very much greater, very much significantly greater for cytochrome C biosensor. So we have developed the cytochrome C biosensors. We have applied the cytochrome C biosensor to measure in cancer cell, to measure the cytochrome C release in cancer cells, and we have followed the cell death. We have followed the cell death by measuring cytochrome C release in cancer cells in this study. In the earlier work, in the earlier research works, we have used platinum electrode as working electrode. Here we have used screen printed modified electrode. The fabrication steps are same. The fabrication steps are same. Pyrrole polymerization, CNT electrode deposition, and CCR bioconjugation. But we have used the screen printed electrodes for cytochrome C biosensor. What are the advantages of screen printed carbon electrode? For example, platinum electrodes are very expensive. Platinum electrodes are very expensive. They are inert, but they are very, very expensive. These electrodes are very, very cheap, and they are disposable. The advantages are we can use only 50 microliter of the sample. For example, biological samples are very, very costly. Enzymes, antibodies are very, very expensive. This is the normal electrochemical cell, and this is the screen printed electrode. And the advantages are volume miniaturization, small size. They are portable. They are portable for point of care applications, low detection limit, no wastage of samples, disposable biosensor, and handheld integration with the microelectronics. In the previous work, we have developed Enzymatic Biosensor. Now we have used the principle of antigen-antibody interactions and developed cytochrome C immunosensor. In this, we have used two platforms, CNT and gold nanoparticle. Instead of CCR, cytochrome C reductase, we have used the cytochrome C antibody as the bioreceptor or biorecognition element. We have characterized the biosensing electrodes using SEM and electrochemical impedance spectroscopy for the immobilization of the antibody and also proteins. Further, electrochemical label-free immunosensor for SOD1 was developed by using immobilizing SOD1 antibody on the electrode surface. So far, we have used antibodies based biosensor, enzymatic biosensor, immunosensors. These antibodies and enzymes are very expensive. So we have used in this work synthetic antibody based sensor. Here, what are the advantages of synthetic antibody based sensor? How they are made? They are obtained from functional monomer polymerization in the presence of template, for example, proteins, small molecules. It is also called molecular imprinting method. The templates are removed to give cavities complementary in shape and geometry and interactions to the template and the protein molecule. So the protein can bind selectively to the cavities for sensing applications. The advantages of MIP based molecular imprinting polymer are low cost. They are very cheap and they are very stable compared to proteins, antibodies. They have long shelf life. Here the probe ferri and ferrous cyanide are used as probe for the analyte determination. I was working in Medical College of Wisconsin. We worked on neurodegenerative disorders. We have found that cysteine levels are altered in Parkinson's diseases, Alzheimer's diseases, ALS diseases. These are all neurodegenerative disorders. In this work, we have developed Cysteine Biosensor using the thiol oxidase activity of SOD1, superoxide dismutase. Superoxide dismutase oxidizes cysteine to give hydrogen peroxide in the presence of oxygen and hydrogen peroxide. The hydrogen peroxide is monitored using horseradish peroxidase enzyme. We have developed and got patents for the Cysteine Biosensor recently. In addition to superoxide dismutase activity, we have earlier studied SOD1 enzyme has got thiol oxidase activity, especially as you see homocysteine, glutathione and cysteine. Only cysteine gives thiol oxidase activity with the superoxide dismutase enzyme. Using the biosensor developed, we have determined the cysteine levels in serum, urine and dietary supplement tablet samples. Here we have measured the values and reported. It was in excellent agreement with the manufacturer reported value. Also, we have developed an electrochemical assay for the measurement of nitric oxide and its metabolites using screen printed modified electrode with the superoxide dismutase enzyme and filed joint patent with the DRDO. This is the commercial available electrochemical analyzer used to perform electrochemical studies, cyclic glutamate studies. The cost of the instrument is now 3 lakhs. Why I am saying this is we have fabricated biosensor electrode and also a biosensor device in our lab. Using NI based myDAQ labVIEW software, we have developed virtual electrochemical analyzer instrumentation for the measurement of various biomarkers to reduce the cost and flexible graphical user interface using labVIEW. This is the virtual electrochemical analyzer developed in our laboratory using the circuit diagram. This is the homemade potentiostat and this is the myDAQ supplied by NI instrument. The cost of this is 25,000. So we have developed virtual electrochemical instrumentation, electrochemical sensor for the cost of 25,000 only. This is the front panel of our virtual electrochemical analyzer, you can see the voltammograms, cyclic voltammograms with the increase in concentration, and the linear calibration plot, and the unknown concentration can be determined by using graphical user interface software. The advantage is that it is used for all the biosensors, whereas glucometer is designed to work only with that electrode. You cannot use the glucometer device for other biosensors. In the case of virtual electrochemical analyzer, we need computer interface to control and measure the biomarkers. So we have proposed a model for microcontroller based portable standalone biosensor device for the measurement of various metabolites, various biomarkers of various diseases. This is the model we have proposed to DRDO, and we have received a grant for developing the standalone biosensor device for various biomarkers. Using microcontroller, we have developed a portable device, biosensor device, for the measurement of biomarkers. What are microcontrollers? Microcontrollers are very small integrated chip, IC chip. It is also present in washing machine, car, TV, cell phone. They can be programmed to control and measure the current response with respect to voltage, sweep. As you see, you can see the cyclic voltammograms. This is a standalone device. Thus, in our lab, we have developed virtual electrochemical instrumentation and also USB interfaced microcontroller based electrochemical biosensor. We have also fabricated in our lab the portable standalone biosensor for high altitude applications. This is the cyclic voltammetric response obtained using commercial instrument. This is the cyclic voltammetric response obtained from our fabricated biosensor microcontroller based device. As you see, they have comparable performance. Here I have combined all my research works and research experiences in the form of a book, Biosensors and Bioelectronics, for the benefit of future generations and students. But the cost of the book is 10,000 because it is published by Elsevier. Also, I have extensively worked on NMR spectroscopy, EPR spectroscopy in my PhD, and also as postdoc research scientist and assistant professor in Medical College of Wisconsin. So I have also combined all my spectroscopy research work carried out in India and abroad in the form of another book, Spin Resonance Spectroscopy, Principles and Applications. It is again published by Elsevier. Further, we have conducted science chemistry exhibition fair to college students and school students. We have carried out more than 45-50 experiments in the fair. I have also combined all these experiments in the form of book, Science Chemistry Fair Experiments for Colleges and Schools, published by our college. I have received more than 1 crore funds from various funding agencies. SERB, DST recently I have received, and the Department of Biotechnology, New Delhi, I have received grants 2 times from Department of Biotechnology, and the DRDO, New Delhi, DRDO, DIPAS, New Delhi, and UGC. I have carried out all my research works with our collaborative researchers. I thank all the collaborative researchers for making the research achievements. Finally, I thank all my research scholars former research scholars and present research scholars for their support and for their hard works. They are now in USA, in SICRI Electrochemical Research Institute, and Switzerland, now in London and USA, and working as faculty members in various colleges. Thank you. Thank you, participants, for keenly listening to my research journey. Hope you have enjoyed the webinar sessions. It is time to thank the principal of the college, the organizers of this webinar, faculty members, for organizing the event very successfully. You will be given e-quiz based on the talks, 15 to 20 questions. The lecture link will be available for you till 5 o'clock for your rewatching and answering the questions. I am also now ready to answer your questions, doubts. Thank you all again for this great opportunity to meet and interact with you through this webinar. Thank you all again.